This method can help answer key questions in the immunology field about antigen immunization-induced T-dependent and independent antibody responses, as well as B-cell activation and germinal center reactions. The main advantage of this technique is that it provides reliable and quantitative Ig isotype response measurements for the direct comparison of B-cell activation and humoral immunity in mice.
For TMP polysaccharide immunization, load 1 ml insulin syringe per mouse with 100 microliters of freshly prepared antigen solution and insert the needle into the lower right quadrant of each animal at an approximately 30-degree angle to avoid lower organ injection. When all the mice have been injected, return the animals to their cages for housing in a specific pathogen-free facility. At the appropriate experimental time points, collect 150 to 200 microliters of blood from each immunized animal by retroorbital bleeding and allow the samples to coagulate at room temperature for 1 to 2 hours. At the end of the incubation, centrifuge the coagulated blood samples and transfer the clear serum from the top of each blood clot into a sterile 1.5 milliliter microcentrifuge tube. After a second centrifugation to remove any residual blood clot, transfer 50 microliters of serum into individual sterile 1.5 milliliter microcentrifuge tubes for storage at -80 degrees Celsius. Before plating, coat individual